Thank you all for participating in this afternoon's media briefing. I'm joined by Dr. Stephen Shapiro, UPMC's Chief Medical and Scientific Officer, Dr. John Mellers, Chief of the Division of Infectious Diseases at UPMC and the University of Pittsburgh, and Dr. Dimitrovic, Director of Pitt's Center for Antibody Therapeutics. They're going to share an exciting scientific breakthrough that is a major step toward a potential drug to both treat and prevent COVID-19. After our experts give their remarks, we'll take your questions. Dr. Shapiro.
Thank you for joining us today to hear about a recent scientific breakthrough that's built on years of groundbreaking research by our colleagues in infectious and immunologic diseases at Pitt, backed by UPMC's clinical, financial, and drug development expertise. Before I introduce the work, I want to take a minute just to recap the unique approach that UPMC has taken to COVID-19 and our reason for holding these regular press briefings. Our basic premise from the beginning is that the most important factor about COVID-19 is the host, the most vulnerable hosts being by far the elderly, as well as those with significant health conditions and the socioeconomically disadvantaged. Masks, distance, and hand washing work really well in preventing spread of the infection. And the UPMC approach has been to protect the vulnerable and to promote this safe behavior. We follow our data, the scientific evidence, and our clinical experience, and we adjust our clinical care and our system-wide preparedness efforts accordingly. And we transparently share these findings as well as our evolving understanding of COVID-19 with all of you. And we did so from the beginning when we developed a test in a week and pivoted our flu surveillance program to track the novel coronavirus, when we provided guidance to federal authorities to protect the most vulnerable and reopen society, when we stood up our tele-ICU not only to our 40 UPMC hospitals, but to our colleagues and patients in New York City, when we launched an international trial using novel clinical approach to test therapies and learn while doing. And as we recently reported, the most effective therapy at our disposal are cheap, readily available steroids. Our analytics has shown that these improved therapies, combined with our clinical experience, likely in addition to some genetic weakening of the virus, have all contributed to the improved outcomes that our patients with COVID-19 continue to have. With respect to specific anti-COVID-19 therapeutic strategies, we've shared with you our published evidence on the immune response generated by Pitt Covac, our vaccine given through a Band-Aid-like patch. And now we will share with you the results published in the leading scientific journal Cell of another potential breakthrough, therapeutic preventive drug that may provide even the most vulnerable hosts with immunity. As Dr. Mellers will explain, there's still much work to be done to make this antibody-based drug available to patients to treat COVID-19, but it's an exciting and promising advance as we will work on multiple strategies to end this pandemic. Let's take a moment to watch this video about the work done by Dr. Mellers and his team, and then Dr. Mellers and Dr. Dimitrov will offer more detail and we'll take questions.
The most cost-effective public health measure is a vaccine. There's no doubt about that. But a vaccine may or not become available in the near future. Antibodies can protect individuals who are susceptible. We've discovered potent human monoclonal antibodies that block the SARS-CoV-2 virus that causes COVID-19. This was discovered in the laboratory of Mitko Dimitrov, and what he does is he displays the antibodies on a different type of virus called a bacteriophage. And he has libraries, many gigantic libraries. These are 100 billion antibodies in a library. And what he does is he takes a protein like that S protein and he pans as if he's panning for gold. He's using the S protein as a bait, and he pulls out the bacteriophage that binds with high affinity to that protein. And he does several rounds of that until he gets the tightest binding protein antibody on the surface of bacteriophage. And then within that bacteriophage, they have the genetic sequence that makes that antibody. And so they can take that sequence, put it into cells, human cells, and produce antibody. We envision two major uses. One is for treatment of people who already have COVID-19. And the idea is that the antibody will block the spread of the virus throughout the body. The other major use is to prevent infection. And some very susceptible populations include the elderly, healthcare workers, individuals who don't have normal immunity, and the idea would be to give a dose of antibody that lasts from weeks to months and protects them from infection. And those who are at risk of acquiring SARS-CoV-2 or COVID-19 could get those antibodies, which would help restore the workforce and get us back on the track to economic recovery. Thank you for joining this afternoon. It's a pleasure to be here. As the video indicated, dating back to February, before COVID-19 was a household name, Mitko and his team of scientists in the Division of Infectious Disease got started on the problem. We're very fortunate, thanks to funding from UPMC and the University of Pittsburgh, to have established a Center for Antibody Therapeutics in Pittsburgh. And we're fortunate to attract my colleague, Dr. Dimitrov here, who has an outstanding record of achievement in finding antibodies to new threatening infectious diseases, including the first SARS. He was the first individual to find antibodies to that, to the Middle East Respiratory Syndrome virus, or MERS, and now to COVID-2. He also developed antibodies that were life-saving for rare infectious disease, highly lethal, caused by the Hendra and Nipro viruses. In record time, back in March, using these billions of antibodies displayed on bacteriophage, Wei Li and Mitko were able to pull out very potent antibodies. And that resulted in AB8 that we've heard about in the Cell publication. What makes AB8 different? It's small, which means it penetrates into areas of the body where a full-sized antibody may not. It's fully human, meaning that there's no foreign material that's likely to be rejected by the host. It's extremely potent, one of the most potent antibodies that's been described. And it appears to be safe, meaning that it does not bind to any human proteins in large screens of human protein libraries. And lastly, it seems to cover all the resistant SARS-CoV-2 variants that have been described in the literature without loss of potency. It's extremely stable when it's produced. The small size means that we can treat or prevent infection in more people with a set amount of protein because we need less of it. And this is extremely potent, and so we should be able to have a greater impact. And we also may be able to deliver the antibody by alternative means than in the vein, which is cumbersome. We may be able to inject it subcutaneously or deliver it by inhalation. And like the COVID vaccine, we're thinking out of the box as to how to improve the treatment and prevention of SARS-CoV-2.
Thank you, Dr. Mellers. Well, we discovered this antibody here in Pittsburgh, it was really our collaboration which made this antibody evaluated and showed this extremely high potency. A team at the University of Texas Medical Branch used this AB8 to evaluate its potency against live virus, and they found that actually at very low concentrations this antibody is extremely potent. Then with this data in hand, our colleagues at the University of North Carolina evaluated the potency of these antibodies in mice, and they found at really very, very low concentrations, like 100 milligram, equivalent to 100 milligram per person, per human, it can prevent infection of mice. And then we not only evaluated this antibody for prevention in mice, but also in another model, in hamster model, and with colleagues from University of Saskatchewan, we found that it's not only very potent for prevention, but also for therapy. When the hamsters are treated, we found that it can eliminate the virus at also very, very low concentrations. So finally, we wanted to know why this antibody is so potent. And then with colleagues from the University of British Columbia, we did this using very sophisticated electron microscopic technique, and we found how exactly it neutralizes and why this is so potent. So I think this team approach to science is critical for addressing the pandemic in the world. And I'm proud that in Pittsburgh we played such a key role, but again, I want to say that the role of our collaborators was critical to evaluate this antibody. And I think that the discovery of this antibody can contribute to our effort to stop this pandemic in the world.
Great. As you can see, this is extraordinary and highly promising work, and it could lead to a drug that both treats and prevents COVID-19, and one that we hope will prove extremely safe and easy to administer. This advance, like all other advances during the pandemic, came quickly, but this was possible because of the groundwork laid long ago by the investments made by UPMC and Pitt. And this new breakthrough is part of an effort that we announced a couple years ago when UPMC committed its initial $200 million to the UPMC Immune Transplant and Therapy Center, or ITTC. It is through this effort that Dr. Mellers was able to recruit his colleague, Dr. Dimitrov, from the NIH, where he led a world-class antibody corps. The goal then, as is now, is to accelerate the development and delivery of life-changing medicine. We're excited to continue backing these efforts to move this work as quickly as possible into clinical trials. Whether it's vaccines, new drugs, more effective COVID-19 treatment protocols backed by our innovative adaptive clinical trials, UPMC and Pitt will remain on the forefront of battling this pandemic and any future threats to the health of the communities we serve. Thank you.
Thank you, Dr. Dimitrov, Dr. Mellers, Dr. Shapiro. And now we'll take questions from the media. To ask a question, you'll have to go to the participants icon at the bottom of the page and press the raise your hand button. That will put you in our queue. I'll read your name and your line will be unmuted for you to ask your question. Again, go to the participants icon at the bottom of the page, press the raise your hand button. That will put you in the queue. After I read your name, I'll unmute your line, but please make sure to unmute it from your end as well. We will try to call on as many different media outlets as we can in the time that we have allotted. Okay, first question is an email question. Dr. Mellers, there have been other monoclonal antibodies mentioned publicly, and some are even in clinical trials. What makes yours so special?
Thanks, Paul. Yes, there are other antibodies that are in development and they're promising. Ours is special because it's extremely potent. And let me just elaborate for a second on what Dr. Dimitrov said about the structure. We know that the spike protein is what facilitates the virus entering cells. And it's in its structure, it has 3 components that are critical for binding to the cell. AB8, through cryo-electron microscopy, was shown to bind each of those components of the spike protein, such that it is likely that variation in one of these will not negate the effect of the antibody, and his team has shown that it's active against a broad range of viral variants. So just to reiterate, it's small, it's potent, it penetrates into tissues, it's fully human, it's active against resistant viruses, and it could be administered by alternative routes, either subcutaneous injection or inhalation.
Thank you. Let's try Max Gomez at WLNY CBS TV in New York. Max, please go ahead. Max, please unmute.
I did.
Okay, here we go. Now
I
we
did.
can hear you. Go ahead, Max.
Rookie mistake, sorry. Dr. Miller's impressive work. I presume then that the binding site is conserved throughout these, through the different strains that you've analyzed. But what I guess everyone else wants to know is what is the clinical trial time frame, and how easily or expensively or otherwise is the AB8 manufactured?
Thank you for that. AB8 should not be a greater expense to produce under CGMP conditions, and we are rapidly moving forward with that effort. Clinical trials require FDA approval, and the timeline, to be brief, is the start of 2021 to manufacture, do safety assessments in other animals, to get FDA approval, and to register and enroll clinical trials.
Next question is emailed to us from Veronica Marshall at Wink TV in Fort Myers, Florida. What's next? Assuming the path forward is smooth and AB8 sails through whatever trials are left with flying colors, what is the timeline for when it could be available to coronavirus patients and the general population?
Well, just to reiterate what I said a moment ago, we're thinking that it will enter humans in the clinical trial in the beginning of 2021. We also have other antibodies related to AB8 that are more traditional size, and they're in production also, and we hope to see those enter clinical trials before the end of the year.
Okay. Let's go to Paul Goff at the Pittsburgh Business Times. Paul, go ahead.
Hi, thanks very much for the time. Can you tell me a little bit more about the Abound, the company that was formed to commercialize this, and also a little bit more about sort of the strategy of that fitting in with all the work that you're doing? And thank you.
So part of the plan of UPMC Enterprise and the ITTC that Dr. Shapiro mentioned was to make the discoveries at the university available for medical use and commercialization. Part of that effort, and a small part of that effort, was to form a company called Abound Bio. UPMC provided seed funding for that. Abound Bio is located in the Hill District, has very energetic and brilliant employees who are working towards the goal of making these innovative antibodies available for treatment of diseases and for commercial potential. Dr. Dimitrov and I are co-founders of Abound Bio.
How about we go to Shannon Perrine at WTAE TV. Shannon, please go ahead. Shannon?
Unmuting. Can you hear me now?
Yeah, we can hear you.
Okay.
Go ahead, Shannon.
Thank you, Paul. Thanks, doctors. I appreciate your time. So for our neighbors here in Pittsburgh, and I understand your information is really going around the world here, but for our neighbors in Pittsburgh, can you explain the difference between a preventative, which is part of this, and a vaccine? This is not a vaccine.
I sure can. Let's just spend a moment differentiating vaccines from antibodies, okay? A vaccine induces antibodies that can prevent infection. A vaccine cannot treat an established infection. An antibody is the consequence of a vaccine, but it's different in that when we give a vaccine, we induce lots of different antibodies of different potency. Here, with this antibody, we're giving a uniform, potent biomolecule that's sole function is to block the virus. So antibody can treat the infection, and it can also, because it's synthetic immunity, prevent infection. And there are populations we're well aware of, particularly the elderly and those with other comorbid conditions, who will not respond well to a vaccine and will need to use an antibody like AB8 to prevent the spread of SARS-CoV-2 in those susceptible individuals. There are also individuals who will react adversely to a vaccine. You all probably know people who can't take a flu shot because of an allergy to eggs or chicken proteins. And for those individuals, a antibody used what we call prophylactically can be very helpful in preventing the infection. I hope that's clear.
Just to jump in there a little bit, so the vaccine, as John said, you take a little piece of the virus or a little protein, part of it sometimes live, attenuated, and you put it in and you ask the host to make an immune response. And that immune response can be very poor, as in the elderly, or very significant, as is in some young people. So getting these things through clinical trials is difficult because everyone's responding a little bit differently. The result, a positive result of this vaccine trial is to make a perfect antibody that will bind to the part of the protein that has been described. And then to take it one step further, because there are some antibodies out there that are going through trials, now you make it 1/10 the size so that it really can penetrate the tissues and get right between the virus and the cell where it enters. And now you have not the Model T antibody, but the Lamborghini, although a really small Lamborghini.
Hey, a related email question, Dr. Shapiro, what's the difference between AB8 and convalescent plasma? And why do we need any of these as a preventative drug if vaccines are going to be available?
Well, as I mentioned, vaccines are going to be available, and we hope that happens soon. But it's more complicated, as we just mentioned. Convalescent plasma is the first start. That's not even the Model T, but we know that some people patients have effectively gotten rid of the virus. So we take their plasma, and in that gamish, there are some antibodies that must work. So that's a start, and the next step is to get just those pure antibodies, which are what's being done with just the monoclonal antibodies. And then, as I said, then you take that and then you make it really small, you make it targeted to the right spot, and then you have AB8. So it's like 3 steps removed and advanced from the convalescent plasma.
Let's go to Megan Schiller at KDKA TV. Megan, please go ahead.
Hi there.
Go ahead, Megan.
Pittsburgh has a history of making medical breakthroughs. The antibody breakthrough put Pittsburgh's medical field once again on the map. Do you think Pittsburgh will also develop the first vaccine?
Will Pittsburgh develop the first vaccine?
We make antibodies, and the vaccine field is quite competitive and crowded. I'm not sure who is going to get across the finish line first, but the real question is not who won the race, but who won the safety and efficacy challenge of developing a vaccine. And so what's most important is what vaccine is easiest to administer, safest, and protects the most people for the lowest cost.
And the Pitt Vaccine Center does have one that has in trials that has been described before, and our Pitt CoVac might take a little bit longer because this is that very novel approach where you put these antigens on a little Band-Aid-like patch. And while that is going to be simpler and actually require probably much less protein and be safer, it's new. So it's going to have to go through more hurdles, but they too are approaching phase 1-2 clinical trials.
And let me just add that competition in science is good. Mitko and I are up all hours of the day, phoning each other, trying to figure out how to improve what we do. He always has the answers, but that's good.
And this requires sharing of information about the structure of the protein. The scientific community has published, 20,000 plus papers in the last 6 months to try and keep ahead of it. The problem with publishing that many papers is not all of them are going to be correct, but we have a lot of information and we're learning a lot every day.
And the competition is really to save lives. That's what it's about. Saving lives by preventing the infection and treating it effectively.
So again, to ask a question, go to the bottom of the page on your screen and press the raise your hand button. That'll put you in the queue. I will read your name, and you'll be unmuted. Please unmute from your end, and we'll go from there. The next question comes from WTHM TV in Harrisburg. Michaela Drapak, please go ahead. Please unmute, Michaela.
Hi, yes. Really quick, we just saw a tweet that says not a single UPMC provider will test a child for COVID unless that child is hospitalized. Can you talk about your reasoning behind that?
I'm not aware of that policy. I mean, we test symptomatic patients and close contacts. We isolate, and if they're symptomatic, we test them. There could be other reasons to test them as well, but we don't have a policy of not testing children.
Yeah, that does make sense. Okay, let's try Bill Kibler at the Altoona Mirror. Bill, please go ahead.
Can you hear me?
Yes, go ahead, Bill.
Regarding all the advantages that Dr. Meller cited for AB8, are there any other antibodies currently in production elsewhere with all those specific advantages?
No, not all the specific advantages. There are antibody cocktails, but this is the smallest sized antibody that is out there.
By tenfold?
In its final configuration, it's about half the size of a full-sized antibody. The key component, the minimum component that neutralizes the virus is about 1/10 the size.
Okay, next question comes from Sarah Bowden, WESA FM. Sarah, please go ahead.
Hi. Yes. So I was wondering about the fact that you have so far only been looking at these antibodies in mice and hamsters, which obviously you have to use non-human subjects before you start clinical trials. But do you think, why are you so optimistic about these findings when you haven't looked at what happens in human subjects yet? Because again, very different creatures, humans, mice, hamsters.
Go ahead. Mitko, please go ahead.
The major reason is our previous experience. We have been developing antibodies against viruses and cancer, and we have antibodies, as Dr. Mellors mentioned, against SARS and also MERS, coronavirus, and Hendra and Nipah. And for example, Hendra and Nipah viruses, they really showed very good neutralizing activity in animals, and then they are tested in humans, and there was a correlation between the activity in animals and in humans. And also records by other groups, for example, the human clinical trial with Ebola virus. So all this previous data tell us that actually testing in good animal models is critical evaluation of those antibodies to predict their efficacy in humans.
And also I should mention that the mouse model was a humanized mouse, so that they put the human ACE2 receptor transgenically expressed in the mouse lung, so that it really is a human antibody reacting against a human receptor. Now, the human obviously has other differences, but just in terms of the efficacy and the mechanism, that should hold.
But the point is well taken from the questioner that the proof is in human studies. But we can show its potency not only in the laboratory dish, but also in 2 different animal models. Same result, very potent blockade of the virus. And we know that we can achieve certain concentrations with dosing in humans. And although there's no guarantee, all signs are pointing towards a positive effect.
We have a follow-up from, I believe, Shannon Perrine. Let's go with Shannon. Shannon, go ahead. Unmute, please, Shannon.
I'm here. Can you put into words the emotions when you were able to isolate this antibody?
So, Miko, what were the emotions you felt when you got the first results of neutralization assay?
I cannot explain in words. It's like these golden moments in the life of scientists when you discover something and you're so excited, so excited. I just can tell you about Wei Li, who indeed discovered the antibodies. He told me that he just cannot sleep for many hours. This is the excitement we have, especially that our antibodies can save lives, as we also demonstrated with our antibodies in the past.
So when you're helping your kids with their science homework virtually, let them know the joy that they will get in life if they continue to do this as a career.
Thank you. Let's go with Max Gomez at WLNY CBS TV in New York. Max, please go ahead. Max, unmute.
Yes, sir, I'm here. I know you said that this was relatively inexpensive, relatively being the operative word, to manufacture, and I know that it's still very early. Do you have any projected cost estimate for the actual final drug?
You know, I think that's a key question, but it's really too early to talk about pricing of a treatment when it's not in humans yet. So unfortunately, I have to defer and say we'll find out. On a positive note, the cost of manufacturing antibodies is falling rapidly as the ability to produce increases. And there are very, very, very few silver linings to COVID, but one of them will be the world will be better prepared to produce biologics like vaccines and antibodies to treat this pandemic and, heaven forbid, the next one. And let me just say that Dr. Dimitrov, working with colleagues, has already identified other coronaviruses in bats that unfortunately can replicate in human lung cells, and he's already identified antibodies that block the horseshoe bat coronavirus, which is the bat that's widely prevalent and has the highest frequency of infection with a coronavirus. So we're not thinking just about this pandemic, but we want to be armed and ready for the next outbreak so that it won't be a pandemic.
Okay, let's go to Veronica Marshall at Wink TV in Fort Myers, Florida. Veronica, please go ahead.
Thank you. You guys mentioned it a little bit earlier, but could you please try to be more explicit in explaining how AB8 interacts with SARS-CoV-2 to treat it and prevent it?
Yes, Dr. Miller just said, we have this electron microscopy pictures which really clearly show that the domain binds to all these 3 protomers of the spike glycoprotein. So this is really a very rare event, and this is how it's one of the mechanisms how it neutralizes very potently. In animals, it's very potent because, as also Dr. Mellors mentioned, because of the small size, it really can penetrate, and we found evidence that the concentrations in the lung and the blood are higher than another full-size antibody. So this is why it's so effective both in vitro and also in animals. Did I answer the question?
Yeah, and just because it has such a long half-life, it can be given before a patient is sick to prevent the disease. But if a patient does have the disease, it can be used to keep the virus from entering the cell once it's already there. So both treats and prevents.
Yeah.
So just to explain a little further in very kind of simple terms, think of the virus as being a burr with spikes on it that stick to your clothes. Those spikes are called the S protein. That binds, those spikes bind to a human protein called ACE2. And what this antibody does is it prevents binding of the spike to the human protein. Think of it as a key and a lock. It prevents the key from getting into the lock, blocking that entryway. So the virus can't infect the cells and cause death of the cell and spread.
Megan Cruz at ABC TV in Orlando, Florida, emailed us this question. It sounds like AB8 is more effective than a vaccine. Can you elaborate in more detail about how it may be better or different?
So we're not making any claims about whether it's better than a vaccine. To defeat this pandemic, we need both vaccines and treatments, and I alluded to that earlier. The most cost-effective means of preventing infectious disease is a vaccine. There's no better example than the eradication from Earth of smallpox, okay, with pennies per dose of a vaccine. Okay, so we're not in competition. We're not trying to be better than a vaccine. As I said earlier, a vaccine cannot treat established infection. An antibody can. And not everybody responds in the way that we want to a vaccine, meaning protection without side effects. And in some individuals, they may get side effects after the first dose and can't get subsequent doses, or they may not respond, and that's where antibodies come in to provide that synthetic protection that doesn't require the host immune system to respond.
And the irony of COVID is we talked about this is a disease of the elderly. The elderly cannot generate, in general, very good immune response. So the patient population that needs the vaccine the most is the least likely to respond. The antibody will provide that for them.
Okay, let's take another email question. This one's from Nicole Brambilla from Public Source. Do members of the research team know about the effectiveness of the antibody treatment should the virus morph as it moves from host to host?
Well, it has morphed. There's one mutation that evolved as the virus moved from China through Europe to the US. That's the so-called D614G mutant. And the antibody we're talking about covers that mutant. Dr. Dimitrov's team looked through the literature and the sequence databases and found other mutations in the region that AB8 binds, made those mutations in the virus spike, and showed in the Cell paper that the antibody was effective against those mutants. So we're optimistic that we'll cover mutants. I've spent my life on antimicrobial resistance. There's no guarantee we will prevent all resistance, but we have a leg up on it to begin with.
And just to be clear, your lock and key analogy, if the virus mutates, it has to mutate that key, so it might not get in that door anyway.
Let's try Colleen Knudsen at WTAJ TV in Johnstown. Colleen, please go ahead. Please unmute, Colleen.
There we go. Can you hear me?
We can hear you, Colleen. Go ahead.
Okay, great. Thank you so much. Good afternoon. So I had a quick question about kind of this fishing and bait analogy that you guys used with the spike protein. And I think in the video it said 100 billion potential candidates were used during that. Can you go into a little bit more detail about that process and what exactly these potential candidates are that you're using?
Yeah, what happens is that we have 100 billion different antibodies which already exist in humans. And then what we do is to use the viral protein, this one, the spike we just discussed, to do so-called panning. Actually, panning, you can imagine that panning compared to screening, when the Westerns, you remember how they get gold out of the billions and billions of sand particles, and they want to find the gold particle, one gold particle. So they do panning. So this is what we do with this 100 billion different antibodies. We want to find one which just binds with very, very, very strongly to the viral protein. And this is the panning. And we can do it in a week, just 1 week, and we have. And this is way, way much faster than other methods which dominate now by using patient serum and then B cells, and then it takes a month to find such kind of antibodies.
Let me just add to that. You may be wondering where the antibodies in these libraries came from. They came from healthy human beings. Many, many donors, their antibody genes were put in to these bacteriophage to make those libraries of antibody. Think of it as a huge library. And what Mitko does is he finds the volume or the book in that library that is key for blocking the SARS virus. He can use that same library to find one to Hendra Nipah or to a cancer cell or many other types of things we want to try to block. He has 10 libraries of 100 billion, so if you do the math, it's 1 trillion antibodies are in his libraries. And he's probably got the largest library of anybody around.
And they're all humans.
And they're all human.
Great. Well, thank you, Dr. Dimitrov, Dr. Mellers, and Dr. Shapiro. That concludes the time we've allotted for today. In about 45 minutes, we will email the media attendees a link to download high-definition footage of the entire press briefing, which we encourage you to use in your media reports. Reporters with follow-up questions are encouraged to contact our UPMC Media Relations Department at upmc.com/media. And as always, please follow @UPMC News on Twitter for frequent updates. Thank you very much.